Hello everyone, and welcome to this talk regarding the use of in silico mutagenesis experiments at a large scale to gain insights into the optimization of the mutational robustness of proteins at various levels. The context of this research considers the well-known central dogma of biology in an evolutionary context. So the flow of information goes from the genotype to the phenotype, which is being selected for throughout evolution. More specifically, DNA sequences are transcribed then translated into protein structures that are characterized by their biophysical properties and their function. When a mutation is introduced either at the DNA level or at the protein level due to translation errors, the effects of that mutation propagate to the phenotype and eventually have an impact on the protein biophysics and/or function, which, depending on the selective pressures, might in turn have an impact on the fitness. The extent to which a given mutation can be tolerated without changing the phenotype is referred to as mutational robustness, and the capacity for the protein to acquire new functions and adapt to the selective pressure is referred to as evolvability. Thus, by studying the impact of mutations on protein phenotype, we can gain insight into the selective pressures and evolutionary mechanisms optimizing mutational robustness and evolvability. These insights can then benefit a wide variety of disciplines, such as protein design, synthetic biology, and phylogenetic inference. So the central question we focused on in this paper is how and to what extent is mutational robustness optimized at various levels of information in globular protein synthesis and evolution. In particular, we limited ourselves to mutational robustness in terms of the impact of mutation on the folding free energy, and we looked at the levels of structures, the genetic code, and codons. So here's the pipeline we used to tackle this question. We first culled the whole protein databank using the PySys culling server with a sequence identity threshold of 95% and a resolution threshold of up to 2.5 angstrom. We then applied our in-house predictor, PopMusicSim, to the remaining structures, and we obtained a mutational stability change estimation for the 19 variants of each residue of each of those structures. So a quick word on the predictor. It basically estimates the Gibbs free energy of folding as a linear combination of knowledge-based statistical potentials evaluated via the inverse Boltzmann law, and the probability of occurrence of a state is sampled as the joint occurrence frequencies of structure and sequence elements in a reference database of X-ray structures. The next step was to map the DNA codons to the protein sequence by first mapping the PDB, UniProt, and EMBL identifiers, and then keeping only the entries that rendered at least 95% sequence identity in an alignment with the translation of the gathered DNA sequences. This enabled us to define ensembles for our mutations at the DNA level, so the ensembles of amino acid variants resulting from single or multiple nucleotide substitutions in a given position of the codon. Finally, we added the codon usage frequencies of the host organisms of each protein by finding their taxonomic identifier and then using the HiveCuts database. So this pipeline left us with a dataset of more than 100 million mutants with known wild-type codon. And in addition, we considered a dataset of 2600 experimentally characterized stability changes for validation purposes, as well as a dataset of experimental fitness change values from 3 different studies. So here's what we found at the structural level. Firstly, residues in the core are less mutationally robust than on the surface. This is well known, and because of the close packing and specific interactions of the core, which tend to impede changes in residue size and physicochemical properties. Note that even if surface residues are more robust in terms of stability, they might not be in terms of fitness, since a lot of them are functional residues. And the experimental data shows the same trend, although it's more noisy. When looking at the relationship between mutational stability change and relative solvent accessibility, we found it to be nonlinear below 20% and linear above that threshold. Moreover, there is a known linear relationship between solvent accessibility and the evolutionary rate, which is defined as the ratio of non-synonymous to synonymous substitutions. So this suggests a linear relationship between stability change and the evolutionary rate, but only for surface residues. For core residues, the stability change decreases faster than the evolutionary rate. And here again, we found a good agreement with the experimental data. Now, protein length and solvent accessibility are somewhat entangled. Indeed, as the protein core and surface are cubic and quadratic functions of its length, respectively, shorter proteins have smaller core-to-surface ratios and thus appear to be more robust on average. Note that this relationship becomes constant above around 400 residues, since these are mostly multi-domain proteins with a constant core-to-surface ratio. Now, since core mutations have larger stability change values, their effect dominates when computing the mean. So if we look further into the length dependence, we actually find that shorter proteins are less robust in the core and more robust on the surface. This is because of the larger compactness and hydrophobicity of the core and the larger fraction of functional residues on the surface of shorter proteins. Indeed, since shorter proteins have less native contacts to compensate for the loss of conformational entropy upon folding, there is a pressure for those contacts to be stronger, and we indeed confirmed an increased content of hydrophobic residues in shorter proteins. Regarding the more robust residues on the surface, those residues are primarily selected for their function and not for stability, which explains why their mutations are less destabilizing. Another interesting result at the structural level is that the proportion of destabilizing, neutral, and stabilizing mutations agrees very nicely with the proportion of deleterious, neutral, and advantageous mutations from the dataset of experimental fitness values. So next we looked at the level of the standard genetic code, and we found that single base substitutions lead on average to less destabilizing variants than multiple base substitutions. This is a reflection of the known fact that amino acids sharing similar biophysical characteristics tend to be encoded by codons that differ by only a single base. This leads to the conclusion that the standard genetic code is at least partially optimized for mutational robustness. Now, one can see that the most change in biophysical characteristics occur for substitutions in position 2 of codons. So we looked at the stability change for substitutions in each codon position separately, and we found that indeed substitutions in position 2 are the most destabilizing, even more so than multiple substitutions in position 1 and 3, while substitutions in position 3 are the least destabilizing. This effect is even stronger when we take synonymous substitutions and the degeneracy of the code into account. So it appears that position 3 is optimized for mutational robustness, but position 2 not so much. However, we also know that position 2 has the lowest rate of translation errors. So we looked at the correlation between this rate and the mutational stability change for all positions, and found an almost perfect Pearson coefficient. So this suggests that the standard genetic code is evolved to optimize mutational robustness against random mutations, yes, but even more so against mutations caused by translation errors. Finally, we looked at the codon usage, which is known to be related to translation efficiency through factors such as transfer RNA abundance, protein expression levels, or messenger RNA stability. So to know whether or not mutational robustness is connected to codon usage, we computed the difference in stability change between mutations in use codons and mutations in their synonymous alternatives, and we found that use codons lead to less destabilizing variants. This difference goes up to 11% of the variants and is present for all three positions, and whether or not we take into account the translation error rates. So this suggests a selective pressure for mutational robustness at the protein level acting on codon usage, especially on the surface where non-functional residues are less constrained than in the core and can thus evolve faster. Here again, the experimental data agrees nicely with our findings. Now, there is also a known codon usage bias at the genome level of most organisms, which has been correlated with the transfer RNA concentration and hypothesized to optimize translation efficiency by tuning elongation speed. So using codon usage frequency tables of the host organisms of our proteins in our dataset, we could show that the increased mutational robustness of used codons versus synonymous mainly happens in biased codons, actually. So, in conclusion, by simply using protein structures and DNA sequences, we were able to quantify the role of mutational robustness at the structural level and its dependence on solvent accessibility and protein length, at the genetic code level depending on codon positions and their respective translation error rates, and at the codon level through codon usage and usage bias. This shows how tools from structural biology can be used in the field of molecular evolution, and it would be interesting to see how other factors than stability contribute to the trends shown here. Thank you very much for your attention.